Imagine placing a living bacterial cell under a bright field microscope. Even though the cell is present on the slide, it appears almost invisible. Why? Because most living cells are transparent and absorb very little light, producing almost no contrast against the bright background. One solution is staining, but staining usually kills the cells and prevents us from observing them in their natural state. So, how can we observe living, unstained microorganisms? The answer is the phase contrast microscope, one of the most important inventions in light microscopy. But before understanding how it works, let's first answer a simple question: why is it called a phase contrast microscope? The name itself explains its principle. It consists of two words, phase and contrast. Contrast means the difference in brightness that allows an object to stand out from its background. Phase refers to the position of one light wave relative to another. A phase contrast microscope converts invisible differences in the phase of light into visible differences in brightness, creating contrast without the need for staining. And that is why it is called a phase contrast microscope. But this leads to another important question: what exactly is phase in light? To answer that, we first need to understand the basic properties of light waves. Although light behaves as both a wave and a particle, phase contrast microscopy relies on its wave nature. We start with crest and trough. A light wave has alternating peaks and valleys. The highest point is called the crest, while the lowest point is called the trough. Wavelength. The distance between two successive crests, or two successive troughs, is known as the wavelength. Amplitude. The height of a wave from its central position to the crest is called the amplitude. A greater amplitude means greater light intensity. See what scientists mean by in phase and out of phase. In phase. When two light waves have their crests and troughs perfectly aligned, they are said to be in phase. In this case, both waves rise and fall together. Out of phase. If one wave is shifted relative to the other, the waves become out of phase. This shift may be 1/4, 1/2, or even a full wavelength. A 1/4 wavelength shift means one wave is displaced by 1/4 of its wavelength. A 1/2 wavelength shift aligns the crest of one wave with the trough of the other, while a full wavelength shift brings both waves back into alignment. But why is this phase difference important? Because when two light waves overlap, they combine to form a new wave. This phenomenon is called interference. It may be of two types: constructive interference. If the waves are in phase, their crests and troughs overlap, increasing the wave's intensity. This is called constructive interference, producing a brighter image. Destructive interference. If the waves are out of phase, the crest of one wave overlaps the trough of the other, causing the waves to cancel each other. This is called destructive interference, resulting in a darker image. Let's see how all the optical components work together inside the microscope. The process begins at the light source, which provides the illumination required to observe the specimen. The light first passes through the condenser annulus. Unlike an ordinary condenser aperture, the condenser annulus is ring-shaped, allowing only a hollow cone of light to pass through. This ring of light is essential because it matches the position of the phase plate located inside the objective lens. Next, the hollow cone of light enters the condenser, which focuses it precisely onto the specimen. Proper focusing ensures that the specimen is illuminated uniformly and that the light reaches the objective in the correct pattern. As the light passes through the specimen, it separates into two components. One portion passes through regions that do not significantly alter its path and continues as undeviated light. The other portion interacts with different cellular structures, changes direction slightly, and becomes deviated or diffracted light. Both of these light rays are then collected by the objective lens. Besides magnifying the image, the objective also contains one of the most important components of this microscope, the phase plate. The phase plate is positioned exactly where the undeviated light passes through the objective. It selectively alters the phase of the undeviated light while allowing the deviated light to pass largely unaffected. This increases the phase difference between the two rays. Now you may wonder why deviated light is largely unaffected but not undeviated. The undeviated light is altered because it passes through the phase ring, which is positioned exactly in its path at the back focal plane of the objective. In contrast, the deviated, diffracted, light travels at different angles and mostly bypasses the phase ring, so it remains largely unaffected. This selective phase shift increases the phase difference between the two rays, creating image contrast. Finally, both rays meet at the image plane, where they interfere with each other. The resulting differences in light intensity create the image, allowing transparent structures that would normally be invisible to appear with clear contrast. Now that we've understood how a phase contrast microscope works, let's look at its major advantages. It allows the observation of living, unstained microorganisms, so cells can be studied in their natural state. It provides excellent contrast for transparent specimens that are difficult to see with a bright field microscope. It is ideal for observing cell division, motility, and other dynamic cellular processes in real time. Despite its advantages, phase contrast microscopy also has a few important limitations. It often produces a halo effect, where bright rings around specimens can obscure fine details. It is not suitable for thick specimens, as excessive light scattering reduces image quality. The microscope is more expensive than a bright field microscope because it requires specialized optical components. Thanks for watching. Like this video and subscribe to my channel.